Hello, everyone. I hope you can hear me. Welcome. Thank you, Mark. Welcome to this Huygens workshop on object analysis. I have two colleagues with me. The guy in the yellow shirt, that is Jasper van Rijst. He will present use case 2 and another presentation he will give. And then Mark, he's also waving his hand. Mark Koenis, he will discuss use case 3. So hello, thank you that you can join this workshop. We are very eager to present our stuff to you. So let's just start. First, I want to go through a few general things. It's probably too late for that, but if you are still interested, register on our website and get an SVI username at svi.nl. And if you do, you can download our software from svi.nl. It's probably too late to also activate your GPU card with the drivers and toolkit that you need to install. So I would skip this for now. Mac users, please visit svi.nl Mac OS for an explanation on how to use Huygens in combination with XQuartz. I hope you all had a chance to download the demo images and the manual. If not, go to the email that we sent to you. And if you did not receive anything, send an email to support and they will provide you with the information that is needed. So in the manual, there is also the login code for accessing Huygens, and I can mention that here as well. So if you start essential or professional, we will demonstrate professional, but the object analyzer is similar in both packages. Login with or workshop user 1, workshop user 2, or workshop user 5. And use the password Huygens_OA_workshop. And if you do, you will be prompted with a license agreement. Just scroll down, hit accept, and then you're ready to go. So if you're stuck, email support@svi.nl and my colleagues can help you with what you want. So please make sure to keep the microphone and camera switched off during the workshop. And I would like to mention a few things to improve the usability of Huygens. If you're colorblind, or if you see that the fonts are too small or the windows are too big, go to the Edit menu at the top in Huygens, select Preferences, then select Display. And if you're colorblind, you can edit the global colors by clicking on that button, and then you select the CBF scheme. So instead of the emission colors or false colors or any of the LUTs, you can select the CBF scheme under the global color mode in Huygens, and then you get the colors that are better recognizable for colorblind people. You can increase the scaling factor. This is for larger monitors. If you lower this, your font will be bigger, and also the window, so you can play a bit with this to get it right for your specific screen. This may need a restart of Huygens, by the way. Now, we have three use cases to present. First one that I will present is about analyzing telomeres within nuclei. So the experiment is confocal data, 3D, of fetal brain cells with telomeres stained with 2 probes, either labeled for Alexa 488 or with Alexa 594. We did restore the image first with Huygens deconvolution and chromatic alignment. So you see on the complete left, the raw confocal data, very noisy, and the deconvolved and chromatically shift-corrected data set. And this we use for the analysis. And this is just an animation of the surface rendering with the nuclei and the telomeres included in it. So we like to thank at this stage the image was provided by Mariette van der Korput from the Erasmus University Medical Center in Rotterdam, here in the Netherlands. And the second case is about the analysis of RNA maturation using mRNA fish. It's a wide field 3D data set with nuclei and 3 and 5 primed ends of mRNA labeled with RNA fish. So we did restore the data set, first with hot pixel correction, then deconvolution, and then chromatic alignment correction. And we cropped the data to make it smaller and suited for most desktop computers, laptops. So we want to thank Mathieu Catala from the RNA degradation group from the Department of Pharmacology and Physiology at the University of Sherbrooke in Canada for providing this data. And the third case is about tracking and analysis of white blood cells attacking fungi. So this was spinning disk data set where white blood cells swarm and invade fungi. It was a 55 gigabyte data set, quite large. The image was deconvolved with Huygens. Mark, who will present this case, stabilized it in time because there was some drift involved, and he did that on the fungi channel. So that's the blue channel in this image. And he cropped the data. So on the right, you see a crop view, and that will be analyzed. And we like to thank Georgios Chamilos from the Fourth Institute of Molecular Biology and Biotechnology from Crete in Greece for providing this data set and allowing us to use it for educational purposes. So the program is in the manual. You can also find it on our website. And the only thing that I think I need to mention is there are 2 short breaks, one of 10 minutes, first one, and that is in about an hour, and the second one that is 5 minutes. You can raise questions during these breaks, and we will monitor the chat box. So if you have questions, enter them in the chat box. And we can also look whether you raise your hand and have questions, and then we can also respond if there is time. Okay, so the object analyzer, let's use it. And for that, I need to open the Huygens software. So I'm just opening Huygens Professional, and I've logged in already as me here at the top right. So if you click here on login, then you will be prompted to enter the username and password, which is then workshop user 125 and Huygens _ OA _ I think it was workshop. So I've opened an image already. This is a stat data set of nuclear pores. You don't have it. Because I first want to show you the object analyzer roughly of how it works, how it is organized. After that, we will do the hands-on with you. So there's enough time to do the hands-on. So I would suggest you just sit back now for a second and relax. We select the thumbnail here, and we go to the analysis menu at the top, and we select here Object Analyzer. I open the image a bit. I try to maximize the window. Okay. So there are a few help functions here to get you started. First, there is an info button here, and this info button tells you in a very short section what this panel is about. So the panel that I've activated is here the primary segmentation group. And you see that here, and here are the functionalities under this segmentation group. So a short explanation. You can click here on I, and that opens the manual with the chapter that is concerned. And there are other options for help under the help menu here. There's an online help function. Also here you can open the manual, and we have a couple of tutorials that are built in this tool. And what I'd like to show you now is the basic tutorial tool. If you've never worked with Huygens before, or reinstalled it, then this is appearing, and it will ask, do you want to be guided by this tutorial? So we just now go through the steps to get a bit of an idea of the Object Analyzer. During this tutorial, it says here the buttons are highlighted in purple that are introduced. So click here to continue. This is what I do. And here it says the Object Analyzer is a tool for segmenting images into objects and computing properties of these objects. On the left, here in gray, you see a whole list of a whole set of panels with different functionalities. In the center, you see a surface rendering of the image. And on the top here, you see some shortcuts that you can customize. So we will all get back to this during the sessions. And here at the bottom, a table will appear with the numbers, with the values that you obtain after analysis. So let's now go to the next step. So if you use the Object Analyzer, your actions will be recorded. And suppose, for example, if we move this scene here, then we can undo this with the button here at the bottom left. Or we can redo the step that we did. So let's undo this, and you see that I go one step back. Then the next session, so segmentation. When you open the Object Analyzer, the first channel of the image is automatically segmented in the primary segmentation pipe. And this works best on deconvolved, restored data. We will also discuss that in this workshop. Then under the primary segmentation group, we go to, just close the info now for a second. We see here the section segmentation, and we see that the stat channel has been opened. So this is just one single channel, and it is stat. So the most important functionality of segmenting data is the threshold slider. And this distinguishes between what is background and what is signal. So if I increase this, I set kind of the background higher, meaning that I get less objects. There is a curved arrow here, which is which resets the segmentation to ODSU. And ODSU is an automatic segmentation algorithm that looks at the intensity distribution, and based on that, it defines a specific threshold value. We can click on these values here and enter them manually if you want to change that. And here below the switchboard, you can click on this button, and we have individual sliders to change the threshold and the seed level. So the seed level indicates, and now I switch back to the histogram, the seed level indicates the voxel value that needs to be reached. So the intensity value that needs to be reached by an object in order to be shown. So this is a second layer of segmentation. And we can, of course, set this back to ODSU again. Okay. Then we have below the threshold and seeding level a minimum object size possibility, and we can define the object size in cubic microns or in voxels. And if I tell the software, for example, that I want to have a minimum object size of 100 voxels, you see that you throw out all the smaller objects below 100 voxels. So we just reset this now to 1 again. Then there is a watershed method, and this can be useful if objects are not properly segmented by thresholding and by seeding. So if we look closer here, sometimes see objects that are just merged, they're not well separated, and we can try using the watershed to improve this. So under the segmentation mode here, under the table where we select the channels, we have here the watershed mode, and we have also an import label image mode, and we also will explain that later on in the workshop. So we select here watershed, and now what we can do is select the color mode, high contrast, and you see that these objects are now separated. We can change the size of the objects with this fragmentation slider by bringing it to the left or the right, and to increase the fragmentation or to lower the fragmentation. We can also apply a Gaussian filter to make objects a bit smoother if they have a rough surface, to also improve the segmentation. Then we can adjust the visualization of the data using this color mode setting. So we have different options here. Uniform means everything becomes one color. And you see in this case that everything becomes green, and that is according to the metadata in Huygens for this specific image. So here it is a green channel. So you see also that the panels are green and the objects are green. Then we can change the colors even with this color bar mode. If we select object label or high contrast, objects of different labels get a different color. And if you hover with your mouse over the objects, you see that they all have a label number. Then with these sliders here, we can change the transparency of the objects. So this is useful if you have a second channel that you want to segment and see that through the first channel. And we can change the brightness here. Okay. Yeah. So let's now change the color mode, not to high contrast or to object label, but to reported parameter. And then we have here a range of options that somehow this window is shown in my second screen. Somehow Zoom doesn't like this. You will see that during the demo yourself if you use it. There's a list of parameters that we can use to color the objects. And one of them is voxel numbers. So here the larger objects are depicted in blue and the smaller objects are depicted in yellow, red. So you have a quick idea of where the large objects are and where the small objects are. Another possibility is, for example, the sum of all voxel values, and this gives a different color distribution. Then we can select objects with these icons here, these mouse icons. And we can select, for example, this magnifying glass, and if we click on an object, we get its properties immediately in the table here below. There are also column mouse modes for selecting regions of interest. For example, this free lasso tool, you can just draw a selection, and we can delete this selection by, for example, going through the object operations and say discard selection. So this situation, we can delete, for example, regions of objects. We can also just group these objects. So there are different possibilities. So let's just now undo this step. Okay. Then we go back to the next stage, object operations. So we did that already. We unselect now for one time the lasso tool. Yeah, okay. Then filtering. So again here, purpley listed. Object operations, filter objects, and we can select here various presets pre-made to discard, for example, large objects, or to discard non-overlapping objects. So we have many different possibilities here, and you can customize things. Then the analysis. So if we go to the analysis menu, here we have options to select as a preset, analysis options, correlation inside channels, correlation between channels if we have multi-channel data, distance to an anchor, distance to a region of interest, and I select now here correlation inside channels. And if I do, and I hit this button, analyze all, I get the distance of each object to its closest neighbor. That is defined in this column here, cm to cm, so center of mass to center of mass of the nearest neighbor. If I hover with my mouse over the table, over the rows, I see that objects are highlighted in the scene, and I can double click on one, and then you see that one highlighted there. And its closest neighbor is about 200 nanometers from the other object. And this is interesting because this is close to the resolution limits of a microscope. We can even sort this column, right click, say increasing. And if we then go to the top of the table, we see that we can separate objects. This is not a fair one because this is segmented. This also with watershed. One I have just two that are quite loose from one another. A bit further. Okay, close enough. Yeah, we're going to the 100, 110 range. Okay, a little bit further away. Okay. Yeah, here we're getting close. So 120, 130 nanometer. Those we can separate already. So that is with STAT. That is, yeah, that's the benefit of using STAT. Okay, let's click further. Okay, in the table, yeah, what we can do is we select a column, and the explanation of the column of what it contains is also listed here in the purple line completely at the bottom. So it says here distance to first neighbor, same segmentation group. Yeah, okay, I need to analyze this all to go through the tutorial to be able to go further. Then the first rows in the table, completely at the top, give you a summary about the image and the conditions of how you segmented data. Yeah. Okay. Then templates. So we can save templates here. If we apply a certain analysis, you can save the template and apply this to other images. And then, okay. Then here at the top, we can customize this row of activities that we want. So if we click on Edit Favorites, we can just drag and drop tiles of certain activities, anchors, for example, defining anchors, or setting a MIP threshold, this is what we can add in this row of activities. Okay, so this is the end of the short tutorial. So let's just now go to the use case with the telomeres. So for that, I open the image telomeres from the use one folder. So you can go to your demo images that you downloaded and select telomeres Alexa 488. You can drag and drop the file into Huygens and then it opens. What you can also do if this doesn't work for Mac, for example, may have a problem with dragging, dropping. Go to the file menu and select open and go to the, with the file manager to the folder and select the image. Now we can increase the size of the thumbnail if it's too small for you with the size button here at the top. For example, you can click on large and the color mode can be set to emission, gray or global. And so if you have that colorblind setting, then this can help with the visualization. Okay, we need to activate one more thing. Okay. Yep. Give me 1 second to prepare. Yep. Okay, so this is an image, confocal data, fetal brain cells with telomeres stained with 2 different probes. And our aim is to analyze the number and the properties of the telomeres within nuclei. So in this demo, we will explore a few solutions to address issues that you are confronted with if you analyze objects within objects. For example, in this demo image, the telomeres may only partially overlap with the nuclei and may be excluded from the analysis. So they're not considered as being in the nuclei because they're just partially in there. So we will try to fix that with the tools in the Object Analyzer. Furthermore, the segmentation of the nuclei may be difficult because they are too close to each other or because they are not homogeneously stained. So let's now see what we can do about this with the Object Analyzer. Select the thumbnail. So you can now work in parallel with me. Go to the Analysis menu and select Object Analyzer. Probably because I have 2 screens, I need to click on the side of the window and in order to see it completely, and then I put it full screen. So again, instant segmentation of the first channel. And it's colored in green according to the metadata. It's also just having one color because the color mode here at the bottom left is set to uniform. So it has just one color. If we would select here high contrast or object label, we see that the objects get different colors. Now the number of objects are instantly shown here below the scene. In this case, there are 125 objects in this channel. But what we want to do now is not selecting channel 0, so the first channel, but selecting channel 1. So we look under the primary segmentation group that is active, under the segmentation tab, that's like halfway on the left, and we select here the orange red channel, confocal Alexa 594. If we select that, we see the nuclei staining, and that is because the background is so high. So if we now grab that threshold slider and put it to the right, we see that telomeres appear. Okay, but we want to make use of the background staining to define the nuclei. So click on that curved arrow to reset the setting to 'Auto'. So So the objects are similarly colored, and we want to threshold them further because we see that there are 3 objects just below the scene, channel 1, 3 objects, but there are clearly more objects, more nuclei. And if we hover with our mouse over these nuclei, we see that these 3 here that are connected all have a label of 1, so they're just one single object. Then here's another one. This is object number 2, and here's object number 3. So to separate these, we use a second layer of segmentation, a third layer of segmentation, I should say, and that is the watershed method. So select from the segmentation mode here, halfway on the left, watershed, and set the fragmentation to 8%. You can also type that in on the left, right of the slider. Then the color mode, we need to set to high contrast in order to see what are now considered as different objects. And also under the scene, you see now that we have 5 objects instead of 3, and the objects even get a different number here. So this is now number 1, number 2, and number 4. So we are now at section number B, because we've defined now the nuclei. Now we want to segment the telomeres. First, in this primary segmentation group that is still active, if you did not click on anything else, change the brightness, that's the complete bottom slider on the left, set the brightness to 0.45 and increase the transparency of the nuclei. So put the slider completely to the right. Then go to the secondary segmentation group at the top left and select there channel 0. So again, you see the green channel, the green objects, and the panels become green. And see again that under the scene, channel 0 has 125 objects. Now move the seed slider, that's the pink slider in the histogram, completely to the left. So we now get more objects, 278. And that is because we also include now objects that have a lower high intensity voxel. Below the visualization panel, we set the color mode to high contrast again, and we see that there are different objects and they are differently colored. Okay, now we set this to reported parameter. That will pop up a drop-down menu, and this drop-down menu, we click on that and we select iParents. You don't see that window with me, but I have. Hopefully you see it as well. So what you now see, if you look at the scene, is that the telomeres get the color according to what parent they belong. And so a parent is a nucleus, and the telomeres belonging to a specific nucleus are colored specifically. So observe that the objects have the same color if they completely overlap with the parent object. Some objects, however, are still gray because they do not completely overlap with the nucleus. Maybe you can see that if you zoom in. What you can do is use the slider just below the scene, grabbing that one and turn it completely to 290 degrees. So we rotate it a bit. Then the twist button here completely at the right of the scene. We can tilt it to 78, 75, something like that. And now you see already that in this red nuclei, there are two objects clearly, three even, that are gray and not considered as being completely in the nucleus. So we can zoom in and out with our middle mouse button, with the scroll wheel, or we can use also the zoom slider here. And if you want to enter a specific value, again, just click on one of the buttons, the zoom or the tilt, and enter the value. To reset the view, go to the top menu, completely at the top of your screen, select options, and almost at the bottom of this menu, select reset view. So we saw that the iParent does not color all the telomeres according to the parent. To include also the ones that are not completely overlapping, go back to that secondary segmentation group and select, instead of iParent, the bottom, which is the parameter for coloring, select now iLabel. Now look again at the scene, and you see that the gray objects in the red nucleus and also in the green one are now all colored. So if there is only a little bit of overlap, voxel overlap, it will now be included in this staining by the iLabel parameter. So set this parameter back to iParent for to work with this, keep working with this. So aside from visualizing the telomeres in the nuclei, we also want to get some numbers, of course. So go to the analysis panel at the left, so the gray panel, click analysis. And now we're going to select a preset. That is not correlation inside channels, which we used previously with the stat image, but we're going to select colocalization intersection. After you do that, click on Analyze All. But I also can show you this. Just above the preset, there is the Experiment Report Editor, and it's also a green button above the scene, Experiment Report Editor. If we click on that, we see a lot more presets. Some basic presets for measuring, for example, do a quick analysis, measuring small particle geometry, correlation inside channels, which we did already on stat data. And we can even customize this all. We selected colocalization intersection, so we hit OK. This was a preset, so now we're ready to analyze all. So click Analyze All, and you get the numbers in the table. So let's now scroll with our mouse button or with the scroll bar here at the complete right to the top of the table. So as mentioned before, the image name shown here in blue and the conditions under which we segmented. The first 5 lines below that represent the nuclei because they have the largest voxel number. 1, 2, 3, 4, 5, and you see that they're boxed in the scene if I hover with my mouse over the specific rows. You also see that they have different labels and that they belong to segmentation group 1, and that this is in fact channel 1, the nuclei. Then in the sum column, we see the sum of the intensities of these different objects. And the iParent row are empty for these 5 because they are parents themselves. On the right of that, that's an interesting field, of course, we see how many children there are in the parents. So in the most right nucleus, we, for example, see that there are 73 children. So if we double click on it, it is highlighted. Then with, if we scroll down, we can look for parent number 2 and click on an object. And we see it also highlighted. So this is a child in parent number 2. So it has 0 as a child, of course, because it's the child itself. So interestingly, you see now here the number of voxels of this object, 314. Doesn't really matter what kind of object you see. The thing is that the colocv column shows the same number of voxels, meaning that all the voxels are overlapping, having colocalization with the nucleus. And on the right of that column, you see the relative colocv, which means the relative colocalization in volume, which is 100%. So that means that 100% of the object is colocalizing with the nucleus. Okay. So now we want to have an overview of how many telomeres are not having a parent. For this, go to the iParent column, click on the top, on the title, iParent. So you see the explanation in the purple line at the bottom of the Object Analyzer. Now right-click and click on Sort Decreasing. Now we go up to the top of the table and we see again that the first 5 rows in the table are the nucleus and nuclei, but after that we see objects that have no parent and no 100% colocalization. So these are the gray objects, in fact. So as we did with the visualization, we could use the iLabel parameter in the experimental report editor to measure, to also include these in the analysis of how many there are in the nucleus. But we're not going to do that right now. We're going to take some different approaches. There are 2 objects here, just below this right cluster, that are also gray. And you see clearly that the nucleus is not extending towards these 2 objects. We could change segmentation of the nuclei by, for example, lowering the threshold and trying to include these objects as well. And I know that will work, but again, we skip this for now and we go to a different option. And the different option, that's session number C, demo cluster analysis. So what we're now going to do is go to the primary segmentation group and unselect the channel here, turn off. So primary segmentation group, turn off under the segmentation panel. So you see now that we only have the secondary segmentation group active with the telomeres. We should have 278 objects. If you do not and want to go along with me, then you may want to quickly restart the Object Analyzer and then activate again under the secondary segmentation group, channel 0, and move the seed slider completely to the left. And if you do that, you're again at the level that I am. Okay, if you're with me, we go to the analysis menu on the left. And under the analysis menu, we select Analyze Clusters. So we're going now to analyze the telomeres based on how far they are from each other and see whether they belong to a cluster. And we do that with the DB-scan algorithm. And this is useful if you, for example, do not have nuclei staining, or if the staining is not of good quality, then you may want to use this type of approach to cluster these objects. There are a few drop-down menus here, 4 in total, to explain what this DB-scan clustering is about. And I would like you to expand the third drop-down menu, explanation of the DB-scan clustering algorithm. And if you do, you see a cartoon. And this cartoon quickly explains of what this is all about, this clustering. Basically, you look at an object and you define a distance in the parameters just at the bottom of this small window. And this distance, this circle or sphere should include at least minimum min point of objects. So we defined here 6 as min points, but in this cartoon, this is 3. So this means that these objects are considered as a cluster with, on top of that, any object that is close to one of these objects in the green sphere, and that is the pink one. So that also belongs to this specific cluster. The one that is encircled gray is not close enough to any other object, so this will be disregarded in a cluster, or it will be it will be discarded for the cluster. Okay, so now click on the checkbox just below the cartoon. Use 2D distance for clustering. And if you hover with your mouse over this title, it gives you an explanation. Check this box to cluster the objects, ignoring the Z component of the distance. So what we do is we consider now that these clusters are not on top of one another in the Z dimension. They're in a flat field of view in XY. That doesn't mean that we need to work with 2D data. This is 3D data, but the clusters are not on top of one another. Then we let the Huygens clustering algorithm, the DB-scan, estimate what the distance is automatically. And you can do that by leaving this to until finish or always. If you hit never, you can enter this value manually, but we rely on the automated estimation. Then the number of min points is 6, and the minimum cluster size is 1. Those are default values, and they work quite well on this data set. If we now click on preview, and since we have this checkbox here checked, color the objects by cluster, you see in the scene, if you move the window a bit away from the scene, that the clusters are colored according, so the objects are colored according to the cluster that they belong to. So I can hit here finish and close, and then these are also analyzed. And I can scroll up in the table, and I can see, for example, at the top, there is one cluster. This is the left one, the right one, the most right one, and it has 79 object counts. So this table has also each object individually shown. So let's just now customize the table. There are 3 buttons at the left of the table, and we select the top one with the wheel. And we can select here, unselect here basically, show object entries. So if we do that, we immediately see in the table that we have 5 clusters, within each cluster quite a number of objects, and this is close to 80, these objects, which is kind of matching the number of telomeres that you would expect in mouse mammalian cells, which have 40 chromosomes, so the double amount of telomeres, which would be 80. but I'm not really sure if this is mouse material. So I don't make a statement on that. Okay. We activate again the show object entries and we click on close. Then scroll down the table and you see that there are still objects that are not included in the cluster. See, there is no cluster index. So the cluster index indicates to which cluster an object belongs, and there's some still empty. And you see that these are also including the two gray ones here. That's this one, line number 279, for example. There are also objects at the top of the image. And let's just now discard these. This is probably just a small part of a cluster that is below the image that we did not collect. So what I would like us to do, and this is session number D in the manual, is pan the image towards that the top right is in the center of the image. And we can do that with our middle mouse button by holding it and moving it around. Or we can select an icon just left of the scene that starts with the magnifying glass. But if we go lower, the third from the bottom, that's two arrows shown as a cross. And if you hover over it, it says pan scene interactively. So if we click that, we can just move the image around. We can zoom in. Let's just now select the cross icon, which is just above that anchor icon. And we can click on objects to simply delete them. So do that for 2 or 3, and then click on undo to let those objects appear again. Now select one of the lasso tools, the free lasso tool or the square or the circle. I take the square and I box select the objects here at the top. And I want to delete those. So I can hit the button delete on my keyboard. I can also go to object operations, which is completely on the left in the panel menu, object operations, and we say discard selection. Okay, if we click again on the scene, then the selection is, the lasso is also removed, and we click on the magnifying glass just to inactivate that selection tool. We can center the image with the square button here at the complete right of the twist slider, just below the scene on the right, to center the image. Okay. Now we want to measure the volume of the clusters. This can be done with a convex hull wrap approach. And this method, this is a known method in which you can consider that we kind of wrap the cluster in plastic foil. We tightly wrap it around the cluster, and we can then measure the enclosed volume for analysis. This approach can be, for example, used if a nucleus staining is very irregular. So we use the pan icon again, or the mouse scroll wheel to center the image, to zoom in or out. And you can reset the view again, just as a reminder, under options at the top, reset view. Now we have the magnifying glass selected. Then we go to the region of interest panel, ROI, expand this one, and click on wrap all clusters. And if we do this, we see that all the clusters are wrapped in red foil. And these are now regions of interest. The panel now shows, or the table now shows these regions of interest. All went well, or am I too far? No, I'm too far. Okay, I should go to the customization of the table. So take the bottom at the top right icon, just left of the table, and click on unselect show object entries. So now we see that there are 4 clusters recognized. So we deleted the one at the top. All again with the different object counts in them. So what we're now going to do is measure the volume of these regions of interest. So we go to the analysis menu and we select small particle geometry from the report preset. So under the analysis menu, we select report preset, small particles geometry. We click on analyze all. And now we have in the table the clusters and the regions of interest. And the regions of interest show the volume in isovolume voxels. So that's in cubic microns. And here's also the surface of the different regions of interest around the clusters. And again here also the object count within these regions of interest is shown. So in this case, this is region of interest number 3, it has 79. And here is the same, but as a cluster, also 79 telomeres. Yeah. Now, there are still those 2 annoying gray objects at the right of this cluster, or just below the cluster that is on the right of the image. So we want to include this in this cluster. And we can do that by selecting them from the table, holding the control button, but that is some work. What we can also do is click on this icon here at the left in the scene, just below the anchor, and it says select a cluster of objects. So if we select that one and go to the right cluster, once it's boxed, we hover over it, it's becoming boxed. If we click on it, it is highlighted. Now we hold the control button and we click on the gray objects. And what we then can do is select see regions of interest, ROI at the left, and then wrap combined. So now you see that the wrap is around the cluster and these 2 objects. And if we now go to analysis and say analyze ROI again, then if we look in the ROI to the object count, we now see that it's 81. So 2 more than the 79 that we had. Let's just now go a few steps back, clicking undo. So 1 step, in fact. So we still have the 2 excluded, and we take now a different approach. We can go to the region of interest panel again on the complete left. Go to the option ROI operations, just above the one at the bottom, which says intensity removal, and we say clear regions of interest. Then what we do is we select the circle selection icon for the mouse at the left of the scene. And we draw a circle around the cluster and the two objects. If we do that, we go back to the ROI menu on the left and say wrap combined. And that is there are 9 different options, and it's the last one in the first section under add to ROI. So ROI, add to ROI, and then wrap combined. And also in this case, you see that it's completely wrapped, and we can analyze, of course, this volume.